but that's fine. EPI can consume those as just additional statistics that you require of the model. All right, quick ellipsis. There's one more step here.
first contributed talk from this session, which is from Virginia Rutten, Alberto Bernacchia, and Guillaume Hennequin, talking about sequential component analysis.
Good afternoon. Everywhere we look in nature, we can see the arrow of time. Indeed, it's rather unusual to see strawberries unrot. Indeed, most natural processes are highly time non-reversible. This could also be said of behavior, be that of animals or of ourselves. So we see that both the sensory input that we receive and the motor output that we exhibit is highly non-reversible. So we might expect that it would be reflected in neural activity. And so as scientists, we might be interested in exploring this link and trying to quantify it. Indeed, when people have looked at neural activity, for instance in C. elegans, they have seen that the neural trajectories underlying behavior often exhibit cyclic trajectories, and that these are explored in one direction and they never come back on themselves. In this specific instance, PCA was used, possibly the simplest thing that you could do, to identify these trajectories. However, a priori, there is no reason why the principal components would be the dimensions along which these interesting and non-reversible dynamics occur. So the question is, how can we identify these non-reversible components? If we look into the literature, there is one recent method that sort of goes along this direction. This method is called JPCA and was developed by Jonathan Cunningham when he was back in the old days doing simple stuff. This method identifies rotational dynamics, and here we can see that in the trajectories of motor cortex, they are indeed very non-reversible. They always go into this counterclockwise direction. However, once again, there's a priori no reason why the dynamics of latent trajectories should be purely rotational. So the question that we ask is, how can we more generally identify non-sequential components in the data? To tackle this challenge, we developed a very simple method that we call sequential component analysis, or SCA for short. If PCA is really the simplest thing that you could do to identify components that capture the maximum variance in your data, I hope to convince you that SCA is the simplest thing that you could do to capture components to capture non-reversibility in your data. So that as soon as you Cosyne ends, if you're interested, you'll be able to carry it out on your own data set and tell us what you find. To explain to you the method, it might be easiest to first go through an example to highlight the difficulties of dealing with sequentiality and how SCA solves them. So say, for instance, that we observe this pattern of activity in trial 1, a traveling wave, this one in trial 2, and so forth. How might we go about describing the sequence of activity? We might say that systematically neuron 1 precedes neuron 2, and neuron 2 precedes neuron 3, and so forth. And we might think of plotting all of the cross covariances across all of the neurons and see that the bump of maximum cross correlation sort of drifts leftwards, indicating this traveling wave. But there's a catch here. Neural activity doesn't typically look like this, but looks more like this. It's noisy. Neurons do not come preordered, and furthermore, there's no reason why the major contributor of non-reversibility be a specific sequence and not just a pattern. And so instead, we're faced with a heterogeneous set of cross covariances, scratching our head at how to find a good ordering of these neurons. So the question is, how can we systematically analyze all of these cross covariances to be able to extract sequential components? In the more general setting, a key insight before that is to note that cross covariances can be decomposed into an odd and even part. If we look across the population, we can see that the even components of all of the cross correlations really capture no information about sequentiality, so we can basically throw them away. In contrast, the odd part really captures this critical information. We can see which neuron leads which. And so we're going to be focusing on this part. In the more general setting, the data might not be temporally stationary, so really what we should be looking is at the full T by T cross-covariance matrices, which can also be decomposed in a symmetric part, and which we don't care about, and an asymmetric part or skew-symmetric component. So what SCA is all about is analyzing the set of the the skew-symmetric part of the cross covariances. So to go back to the method, we have our data. I'm going to reconstitute all of these skew-symmetric matrices into one large matrix, which itself is symmetric and thus is real and has orthogonal eigenvectors. Next, what we're going to do is we're going to do eigendecomposition of this. Now, given the size of this matrix, which is nt by nt, n being the number of neurons and t being the number of time bits, this would be horrendously expensive. However, by exploiting the structure of this matrix, we were able to derive an efficient iterative algorithm to be able to do eigendecomposition of this matrix such a way that it scales much better than the naive approach. So then we can look at the top eigenvectors associated with the largest eigenvalues, and these are what we're going to call our sequential modes that we can reshape as an n by t matrix. Just as in PCA, we then project our data onto these modes, we can do the same thing with our sequential modes. And we'll do that a bit in a short moment on some real data. But sometimes what we're really interested in is looking at the trajectories, the time course of neural activity in some low-dimensional space. We can also do this in the SCA framework. Instead, if you want to do this, then we're going to extract the top spatial subspace of the matrix that we call CSEC, and which captures the maximum power in this matrix. Then we will have spatial modes that, once again, a bit like PCs, we can project our data on and find latent trajectories. So for our tau data, for instance, if I project my data onto our top modes, we find that the hidden latents actually look like this, where all of the dots are the starting points, all of the rotations were in the same direction, so it was really sequential because that's what the method extracts, and they're just sort of interspersed for clarity. In contrast, if I apply PCA, this is what it looks like. For the simple reason that in another latent subplane, there was a large amount of non-reversible noise, highlighting that whereas PCA really picks up on this noise, which is often more reversible than other sort of signal dynamics, SCA is completely immune to this, showing its denoising capacity. Okay, so now that we have SCA, we were eager to apply this to some real data to really put it to the test. We analyzed some data from rat hippocampus that was generously shared to us by Brian Lustig, who works at Janelia in the Albert Lee lab. In Brian's task, the rats at the start of each trial have to select one of 3 arms of a maze and has to do this in a specific sequence. In between each of the trials, the animal stays in a delay area and has a timeout period during 8 seconds, during which he has to run in a wheel. We specifically analyzed this, the activity during this delay area. This is what the sequential modes looks like. They look sequential, more or less. And sorry, I forgot to mention that, of course, any of the skeptics among you might say, well, given enough neurons, I can always make something look like a sequence. But there's a way of distinguishing how many sequential modes really contribute to your data by using a bootstrapping method to estimate a noise baseline. So these modes were significantly above that. Next, I can project each trial onto these top sequential modes or principal component modes. So each dot here is indicating one of the trial, and we see that the top 2 STs capture more of the behavioral relevant conditions in the trial, while the animal is always doing the same motor activity, indicating or sort of suggesting, going in the right direction, that sequential components might indeed carry more behavioral relevant information. We believe that beyond the hippocampus, there's potentially multiple applications of SCA, and so if you're interested, please try, tell us what you find, or get in touch if you have any questions. Before sort of leaving, there's another area which we think that thinking about non-reversibility might be interesting. In statistical analysis of neural data, we can traditionally divide it into 2 sets. One that looks more at the dynamics, such as JPCA, versus non-parametric Bayesian approaches. Both have their benefits and complementary pitfalls, but there's really sort of nothing in between. Working on SCA, we reasoned that often dynamical systems exhibit very non-reversible dynamics by the fact that they follow a consistent flow field. However, when you look at the sort of priors that are used in Bayesian parametric, non-parametric methods, often these assume fully reversible trajectories. So we thought that maybe we could add in this non-reversible component and perhaps sort of bring together the two branches slightly better. To do this, let me go back to JPCA to sort of, work us through the idea. So in JPCA, we fit a linear dynamical model which is constrained to find rotational dynamics. In fitting this dynamical model, we identify a latent subspace in which the dynamics rotate. However, we could do away with the dynamics and instead write the analytical form of the space-time covariance matrix and then use this as a kernel and impose a rotational prior onto our latents, thereby reinterpreting JPCA as maximizing the marginal likelihood of our data under this simple probabilistic model. Why do this? It has multiple advantages over the traditional JPCA, for instance, by giving you uncertainty estimates or allowing you to gracefully deal with missing data if neurons drop out during a recording, for instance. So here, for instance, we have a GP running in the latents, and then you have observations via linear readout. Sort of in parallel, conversely, we can think of bringing GPFA, closer to the dynamics approach. By generalizing the kernel that we had that is purely rotational and using some of the maths that we derived using SCA, we could generalize this rotational kernel to one which only assumes two things. One, that the activity trajectories are smooth and that they are irreversible. So on one side, to compare, we have purely squared exponential reversible dynamics, and on the other side, we have dynamics drawn from this new kernel. And you can see overall that there's a consistent trajectory rotation, more or less, in our kernel. It rarely falls back on itself, but that overall it still displays sort of diverse broad set of trajectories. And we think that this might be a better prior to have when fitting models such as GPFA, as it better reflects our own prior about behavior and neural activity. To conclude, SCA is a very, very simple method which scales well to large data sets, allows you to extract sequential components of neural data, and gives you both a spatial dimensionality decomposition of your data as well as a spatial dimensionality reduction technique, and potentially gives us new promising approaches to bridge slightly more the gap between dynamics approach and Bayesian approach. With this, I'd like to say a big thank you to the team. I wouldn't be without them, to an incredible mentor, Guillaume Hennequin, Alberto, who's a real whiz, and a very generous Brian Lustig and talented. Thank you very much.
So we have time for a couple of quick questions.
Hi. I'm not sure I understood what you mean by reversibility in this context.
So ideally, I would have a backup slide, but I don't have that for this. So reversibility, by this we define it that the probability of the covariance between neuron 1 at time 1 and neuron 2 at time 2 be different than the probability of neuron 1 at time 2 and neuron 2 at time 1. So it's not so it's only up to second order, our analysis, by the fact that it's only analyzing covariances, only considers second-order statistics, and we're not saying that something is reversible or non-reversible. There's a degree of reversibility, and that's also something that we're exploring in the kernel, is that we can add an extra hyperparameter alpha, which allows you to scale, smoothly between a purely squared exponential kernel and a purely sort of purely reversible and purely non-reversible, and sort of have a spectrum. So ideally, you'd also be able to optimize this parameter.
Hi, great talk. Should we be thinking about the modes with the largest eigenvalues as still like the highest variance irreversible ones, or like the most irreversible? Like, are they the most sequential, or are they still just like the highest variance, but still irreversible?
So I would interpret that as carrying the most variant of the non-sequential, not that they are the most non-sequential. Another note on that, another distinction that I didn't make between JPCA, for instance, is that JPCA, the top eigenvalue is actually just going to be the largest mode. It's not necessarily the cleanest plane in which the rotations are, and that's a real benefit of using this more GP-based JPCA, as the rotational plane that you'll find will sort of be the cleanest or the most suitable under the log likelihood.
So I think we're running out of time. Sorry. So we're going to have to switch gears. Let's thank Virginia again. Right. So we are continuing with Cody Baker, who's going to talk about nonlinear computations in semi-balanced networks.
Okay, thank you all for coming to my talk today. The work you're about to see was developed by the Rosenbaum lab at the University of Notre Dame, in particular graduate students myself and Vicky Zhu, and supported by multiple NSF grants, including NeuroNext. So there's a notion of approximate excitatory-inhibitory balance that has been widely observed experimentally in cortex. And one of the questions our lab asks is, how does this notion of balance shape the functional properties of neural responses? And of course, what do I mean by balance? A quick mathematical review. All I'm saying is the total input current going into a network of neurons can be decomposed into two sources, a strong source of recurrent input and a strong source of external stimuli. And each of those can be scaled together by the average coupling strength in the network, JK bar. And balance at this level essentially becomes synonymous with the cancellation between these sources. And that cancellation gives rise to an analytical expression for the firing rates of neurons in such a system as a one-to-one function of the synaptic parameters W and some external stimuli X. Now, this equation has been used already to study all sorts of wonderful, interesting properties in cortical circuits. However, it has some issues. Primary to the talk I'm giving today, you'll notice it's a linear function of stimuli. But we know that our brains are capable of performing nonlinear computations every day of our lives. So that just means we can't use the classical balance framework to explain that. Furthermore, the synaptic parameters actually have to be precisely tuned oftentimes to avoid producing negative rates in the expression, and that just gets worse the more subpopulations or cell types that you consider. Further, we were actually able to prove that for any specific network satisfying Dale's law, you can always find some sort of external input pattern that can produce negative entries in the expression. And so to illustrate these ideas in a simple model, I'm going to consider two external populations and two excitatory populations in a total recurrent network of ADX neurons. And as we transition between two types of stimuli, we're going to start in what looks like the classic balanced state. We have strong excitation, strong inhibition, but there's some total cancels out to something very, very small over time. And this gives rise to the nice asynchronous irregular firing patterns that we see in those raster plots. But as we transition to a new type of stimulus, we notice that we've broken balance in the first excitatory population. In particular, the sum total input current is now hovering on a very strong negative value, and by consequence, the neurons in that population have stopped spiking. They've become completely silent. But interestingly, you'll notice the remaining part of the network has rebalanced itself. There's a slight amplification to the firing rates in the E2 population, but that may simply be to offset the silence in E1. These two properties together define what we're going to refer to from here on out as the semi-balanced state, where some populations are receiving excess inhibition that would originally be considered breaking balance, but the remaining subpopulations in the network form a balanced subnetwork. So the question now is, how does this notion of semi-balance shape neural responses? Well, if we go back and rederive the rate expressions under these new assumptions in the strong coupling limit, we obtain an implicit equation for the firing rates written in terms of a rectified linear function. And a rectified linear you can think of being like the simplest type of nonlinearity that is, for instance, often considered as an activation function in artificial neural networks. And this gives us now a nice nonlinear mapping between the stimuli and the rates. And furthermore, it can be mapped directly onto fixed point rates in recurrent neural network models like echo states that use ReLU activations. And also the threshold linear network studied by Karina Kurto, who gave a talk on those at this very conference 2 years ago. And even though the exact expression is derived in the strong coupling limit, it nonetheless provides a good approximation for networks with biologically realistic coupling strengths, like those derived in the recent Ahmadian and Miller paper. And so returning back to our simple example of the multiple populations, we test the efficacy of this theory in the dashed line and see very good agreement between the empirically observed firing rates in the solid lines. But more interesting than just testing the theory, you can actually take a linear readout from this kind of system and very easily shape it to produce a continuous XOR operator, which you can again think of as being like the simplest type of example of nonlinear computation. It's one of the simplest types of problems that no linear separator can solve. Okay, so far I've only talked about all this stuff as occurring at the level of averages over multiple populations of neurons. We can extend this much further down to the level of individual neurons, the so-called detailed level. And the notion of balance in this situation now is considered having to do with the diffusion or the variability of the individual sum total currents around their average. So you see in our quote-unquote detailed imbalance state, there's a pretty wide distribution of the total input currents in black. That is to say, no individual neuron is ever per se precisely balanced, and this gives rise to a very heavy-tailed distribution on the firing rates. But there is a well-known property of a homeostatic inhibitory spike timing dependent plasticity rule developed by Vogel back in 2011 that actually enables detailed balance to occur. And you'll see within a few seconds of simulation time upon activation, all of the sum total input currents for the neurons in the network converge around a very highly concentrated value. And by consequence, the firing rates all tend to hover around a very central average. Okay, but so far I've only talked about time constant stimuli, and that's how all of this is achieved. What happens when we consider time-dependent stimuli, in particular ones that evolve faster than the plasticity rule can actually keep up with rebalancing things? In this case, we observe rise very naturally all of the properties that I had previously mentioned for the semi-balanced state. You see that balance is broken, but only ever in the negative direction, corresponding to mixed intercombinations of neurons becoming silent or amplified. And so you can tap into the inner workings of this model and plot a joint distribution of multiple firing rates as the parameters of the stimulus are varied. And these form some very rich nonlinear manifolds that end up being very useful for performing almost arbitrary nonlinear computations, even by doing something like a simple linear regression of points on the manifolds. And to highlight how this could be used, we used a very common popular machine learning data set, the MNIST handwritten digit data set. If you simply feed these images into our networks and obtain semi-balance, then you'll find, or at least what we found over 2,000 randomly selected images, is that those images were not linearly separable within the pixel space, but they were perfectly linearly separable in the rate space, at least over the training set. But it did generalize fairly well, even if you consider a biologically realistic case of spiking output neurons following Dale's law and just using a simple supervised Hebbian plasticity world to train them. Okay, there's many more wonderful details on this in our preprint that's on bioRxiv under this name. Quick plug, as of last week I had my PhD, so I'm looking for work in industry positions related to comp neuro, data science, machine learning. You know of any, add me on LinkedIn or shoot me an email. Thank you for listening. Are there any questions?
Okay, so we have time for some questions, and maybe the next speaker can come and set up while we're doing that. So I guess I have to start. So I was wondering, can you do temporary dynamic processing with this framework, or sort of you're treating the inputs as stationary data points?
So you're asking if we tried to regress upon a time series?
Yeah,
as our
sort of
goal.
like if your inputs are time series and you're trying to do classification or sort of like representation in that space.
Static classification, that is essentially what we did with, for example, AIMNIS and even the previous examples. The way we obtain the time-dependent part of the stimulus is by fast switching. We have not tried to do actual regression upon a smoothly varying time series, but we have gotten that question before.
Okay, so and you have some notion about how the spectrum of the input gets processed through this filter?
So that comes down really relative to the time scale of the plasticity, which has to be tuned somewhat precisely relative to the stimulus in order to produce a semi-balanced state. That is something we have not quite, certainly not analytically worked out, but it's certainly one of the open questions. Yeah.
Okay, more questions?
Yeah, just a question with regard to this last result. So even in a strictly balanced network, the individual neurons are still nonlinear. So would this result not be possible in a strictly balanced set?
That's true. I mean, you can use a classic balanced network, balance at the mean field level. You can still regress upon it, and you're right, the individual activations are still nonlinear in some fashion. But we've always found better performance using the homeostatic plasticity to really enforce the notion of the semi-balance. But yeah, that is a good point.
We have time for one more question.
Okay, yeah, thanks for your talk. Simple question. What did you mean when you said the balance was only broken in the negative direction?
In the negative direction, being excess inhibition. So there's no balance broken in the excitatory direction. That would have led to classic balance being broken and the theory not working out at all. Mostly highly amplified spiking rates.
Okay, thanks.
Okay, so let's thank our speaker again.
It is my great pleasure to be here with you today to tell you a little bit about the work that I've been doing in the Clark Lab at Yale. Motion perception is crucial for survival. It is crucial for a lioness hunting its prey, who's trying to elegantly get away. It is crucial for us trying to cross a busy street where you have multiple vehicles coming at us at different speeds and from varying distances. So given how crucial motion perception is, it's perhaps a bit surprising that the brain circuitry that we have to do this motion computations sometimes makes mistakes. And we see motion in patterns like this one, where it is in fact no motion at all. And I invite you now to experience that illusion of motion. Let's see if this works. This is a very large auditorium, so doing psychophysics in such conditions is, is bound for disaster. But let's try. Just look in my direction, and I'll flash this pattern in hopefully the visual periphery of some of you. Here we go. Look in my direction. What I hope you should be seeing while the pattern is on the screen is slow clockwise rotation of the pattern. Maybe some of you saw it. It's hard, but it is in fact true. So this animation, and I'll show you data. This animation actually contains another illusion. This one is easier to see if you look straight at a pattern this time around. And you should see anti-clockwise rotation of the pattern as the pattern disappears. And this is quite a bit stronger. Something that is interesting about illusory motion from stationary patterns is that other animals also see it. For example, this cat, it's looking at a pattern that is related to the one I just showed you. And it certainly seems like it's seeing something is moving and it's trying to catch it. And this is anecdotal, but there is indeed evidence that other vertebrates also see motion in patterns that are stationary. We want to understand the mechanisms for this. So we want to ask, first thing, do fruit flies also see motion in stationary patterns? If they do, first we have a lovely way of measuring behavior. And secondly, with vast array of genetic tools that exist to manipulate and measure specific cell types in the brain of the fly, then we really have a good chance at understanding this illusion. So how do we know if the flies see the illusion? Well, we'll look at their behavior while we show them a modified version of the stimulus. What in particular about their stimulus? Well, there's something that is known for a very long time in the field. If you rotate a pattern around the version of the pattern that they prefer, a stationary version of the moving stimulus that they prefer. And the same thing happens for the other pair. So how do we put these things together? So we have a behavior, we have the responses from these neurons. How can we understand the behavior from the responses of these neurons? Well, one simple model tells us that the direction in which flies turn is dictated by the sign of the subtraction of the activity from B neurons from the activity of A neurons. If we apply this to our stationary pattern, these are the two neurons that are most involved in responding to this pattern. And we know from our behavioral measurements that this behavior is positive, so flies turn towards the right, which suggests that the T4A neurons contribute more to this behavior than T5B neurons, or more generally, that T4 neurons' contribution to behavior outweighs that of T5 neurons. So T4, to remind you, are the on-motion detectors, and T5 are the off-motion detectors. And you'll also notice that T4A and T5B are opposite in two ways. One of them responds preferentially to motion to the right, the other one to motion to the left. One is an on-motion detector, the other is an off-motion detector. And what this model also allows us to do is to plan how we can manipulate this illusion. It suggests that it should be possible to manipulate the illusion. For example, if we silence T5, now T4A is the neuron solely responsible for controlling this behavior. So fly should now turn in the same direction as before, but stronger. And if we silence T4 neurons, now T4B will be the only neuron controlling this behavior, and we should see behavior in the opposite, negative direction. And that's precisely what we found. If we silence T4 neurons, now flies turn in the same direction as before, in the same direction as controls, but slightly stronger. And if we silence the T4 neurons, now flies start seeing motion in a stationary pattern in the opposite direction than what they used to. So this is pretty awesome. We've really nailed down a causal mechanism for this illusion in flies. So to explain this illusion, we need three things. We need direction-selective neurons that respond to stationary patterns. We need opponent subtraction of left and right signals. And we need different weightings for light and dark moving edges. None of these is particularly fly-specific, so we thought maybe it's not completely nuts to hypothesize that this could also be the basis for the illusion in humans. So we decided to test that. We asked folks to look at this pattern to tell us, when it's stationary, do you see motion clockwise or counterclockwise? Then we added physical motion to the pattern in one direction or the other and at different speeds until we found the velocity of the pattern that cancels out the illusion of motion in each person. And that's called the illusory velocity, that velocity. This is what we found. This is the range of illusory velocities we found for our 11 participants. And I'll remind you that in flies, the reason why we concluded that on and off asymmetric weighting onto behavior must be involved in this illusion is that when we silenced the on pathway or silenced the off pathway, we were able to manipulate the strength and the direction of the illusion. Can we do something similar with flies? Well, we can't silence neurons in flies, but we can show visual adapters, right? We can do light edge adaptation to decrease the gain of responses in the brain of humans to light edges, or show dark edges in both directions, so there's no some after effects, to decrease the gain of light edge responsive neurons in the human brain. And so that's what happened. This is what happened when we show light edges to people. We see that the illusory velocity goes down, and in fact, now more than half of the participants see the illusion in the opposite direction, just like we saw in flies. But it will come as no surprise for anyone in this audience that the brain of the human is more complicated than the brain of the fly. And so when we use the dark edge adaptation, contrary to what our model predicted, the illusory velocity also went down. But what I would like you to pay attention is that despite not being fully predicted by our model, the responses, the consequences of light edge and dark edge adaptation to the illusion were different, and also that these responses are consistent with a model in humans where light and dark edges could be at the cause of this illusion. In summary, I've shown you that flies see motion in stationary patterns like we do, like other vertebrates do. We found a causal mechanism for this illusion. It involves motion detectors. We were able to enhance or invert the fly's perception of motion, and we did this in humans as well. And using human psychophysics supports a similar model. With that, I would like to acknowledge the Clark Lab at Yale, in particular my advisor, Damon Clark, my co-authors on this study, Ryosuke and Emilio, and I invite you to check out Ryosuke's poster on Friday. Thank you very much.
So we have time for questions. Also, could the third speaker come up? We need your signature. You got it.
Hi, I have two questions. First one is you mentioned that the weights for T4 neurons should be bigger than for T5. Is that consistent with like previous studies? Because I feel like the off signal is like more important for flies.
The off signal is more important? How do you mean?
Like the L2 directed information is generally more than the L1 directed on information.
So I don't mean necessarily that the responses of T4 are larger than responses of T5. I'm saying that whatever happens downstream, the weighting on downstream neurons for T4 and T5 is different. And that's what come out of our results. Our results can only be explained if T4 contributes more to behavior than T5.
Okay, another question I have is, when you measure the neuron response, it seems like it's a transient response.
It's a transient response.
Yeah.
Oh, well, the x-axis was not time, was space.
Okay. So when you present the stimuli to fly, and they can actually turn for as long as 5 seconds?
That's correct.
Yeah, but...
Well, but so if you look at each individual fly, they don't consistently, this is on average a lot, maybe 20 flies. So, but there's certainly a bias for each fly to turn to the right. But they wiggle a bit, yeah.
So you mean the neural activity versus the behavior, they are like on for similar temperament?
Yeah, there's a match. Yeah, especially for T5 neurons we found. They are pretty sustained. T4 a bit more transient. Yeah, thank you.
Thank you.
I was just wondering about phase invariance in the fly visual processing, like the neurons that you're interrogating here, because I wonder if the difference between human behavior and the fly behavior has something to do with a difference in phase invariance in the two visual systems.
I think that's one key. And
Okay.
we don't know the answer. But
Okay.
what we thought we could do is, take so things that are different between human and flies are a ton. Two of them is that flies have one neuron that responds to on edges, one neuron that responds to off edges. Certainly not the case in humans. Humans have many spatial frequencies, many temporal frequencies. So our guess is that we put together a motion energy model with a bank of filters that we would find. Maybe we'll be able to explain the difference.
Mm-hmm.
Do you know why we have the difference between the light edge, dark edge imbalance to begin with?
Whys are difficult, but what we do know, first, there are other studies that indicate similar results, but we do know that natural images have an asymmetry between light and dark. So,
So embedded in the natural image statistics?
Exactly.
All right, let's thank Margarida again.
Yep.
Okay, yeah, our final talk of the session is from Ni Ji and is called Sensory Control of Neuromodulation Enables Flexible Control of Behavioral States.
Organizers for this great opportunity, and thank you all for being here. So a common feature of animal behavior is its ability to be both stable and flexible. For example, city squirrels foraging in the park are often seen consuming human food with great focus. However, it will not hesitate to explore new options if you present it with some exotic new food variety. The ability for foraging as well as for many other behavior contexts, the ability to sustain a behavior state, as well as the flexibility to switch between alternative states are both important for survival. The question is, how does the brain achieve both? In particular, we would like to identify general neural circuit architectures that could give rise to both stability and flexibility. So we sought answers to these questions in the simple brain of C. elegans. Just like many other animals, C. elegans alternate between exploration and exploitation when they forage for food. In the roaming or the exploration state, the animals travel persistently at high speed, sampling a large area of its habitat. In the dwelling or exploitation state, the animals suppress its locomotion and focuses on consuming the food at hand. On a uniform environment, the transitions between these two states appear stochastic. However, these transition probabilities can be readily modified when the foraging environment changes. So a previous study has identified two neuromodulators, serotonin and PDF, that respectively promote the dwelling and the roaming states by targeting a set of downstream neurons. In order to understand how these neurons function together in the circuit to control the foraging states, we have developed technology to perform ensemble Cosyne imaging in a freely foraging animal. And by aligning circuit-level neural activity with behavior, we were able to identify a low-dimensional representation of both the locomotory parameters and thereby the foraging states of the animal. In other words, there is a robust low-dimensional representation of the foraging states. Two additional features of the circuit-level activity also caught our eyes. First, we observed persistent neural activity in many neurons, most notably in the serotonin neuron, whose activity tends to span the entire duration of the dwelling state. Secondly, we also noticed mutual exclusivity between the activity of the serotonin neuron and that of the neuron that releases the neuropeptide PDF. And their mutually exclusive pattern maps well onto the roaming and dwelling states. So this led us to naturally ask, what is the functional circuit that gave rise to these activity patterns? So through, we combined genetic perturbations with circuit-level Cosyne imaging, which led us to identify a mutual inhibitory circuit that links the serotonin neuron with the neurons that are either targeted by serotonin or the PDF. And consistent with this functional circuit topology, blocking serotonin signaling led to the shortening of the activity duration of the serotonin neuron itself. Disrupting PDF signaling had the opposite effect of prolonging the persistent activity of the serotonin neuron. In addition, blocking PDF signaling also disrupted the mutually exclusive activity pattern seen in the wild type and resulted in a foraging behavior that's somewhere in between the roaming and dwelling state. So these results together reveal that this mutual inhibitory circuit critically gave rise to the bistability observed both at the neural circuit level and at the behavior level. And this is consistent with theoretical predictions from a long-standing series of work in the past. So we next wondered, how does this mutually exclusive circuit flexibly control its output state? Interestingly, we noted that several of the neurons that are targeted by serotonin and PDF are well known to be involved in either sensory processing or motor control. So this led us to hypothesize that maybe the circuit dynamics within the sensory motor pathway could impact the propensity of this circuit to switch between states. And if that is true, then we should be able to glean at the ongoing circuit activity and be able to predict circuit state transition at the next time window. So in order to test if that is indeed the case, we adopted a popular method in the machine learning field, which is the convolutional neural network method. And we use this method to predict circuit state transition from the ongoing circuit activity. And using this approach, we were able to consistently train neural networks that achieve over 70% test accuracy. So among the trained neural networks, we then pulled and clustered the first layer convolutional filters in order to identify common filters that are shared across networks. And shown here is one such convolutional filter. And the important point is that the weights on the filter correspond to individual neurons, the activity of individual neurons at different time windows. Then next, through feature selection, we focus our attention on the sensory processing neuron, AIA, whose activity is singly indispensable for the classifier performance. So to test whether this neuron actually plays a role in influencing state transition in the circuit, we then optogenetically activate this neuron in foraging animals. Upon stimulation, roaming animals rapidly enter dwelling state, resulting in a sharp drop in their average speed. The probability to transition from dwelling to roaming also increased, although at a slower time scale. These results suggest that AIA might provide excitatory drive onto both the serotonin neuron and the neurons that are targeted of the PDF signaling. Then by examining the C. elegans connectome, we also know that AIA receives convergent sensory input from multiple sensory neurons. And this led us to hypothesize that maybe AIA is able to couple circuit state transitions with dynamic input from the sensory environment. So in order to test this hypothesis, we presented foraging animals with a heterogeneous environment where there's a denser food patch some distance away. And food-related odors emanating from this denser food patch could then help these animals navigate up the gradient to eventually arrive at the better food source. So these animals are indeed able to achieve this, and they seem to do this in two ways. So first, they can increase their time span in the roaming state, which allows them to travel afar and eventually reach their destination. Secondly, they seem to be able to carefully time their state transition such that they preferentially switch from roaming to dwelling when their direction of motion start to deviate from the gradient direction. And most importantly, both of these foraging strategies require both AIA and the sensory neurons, which again suggests that AIA plays an important role in coupling dynamic sensory input to circuit state transition. So we wanted to get some intuition on how this circuit might be able to achieve this flexible control of persistent behavior states. So for that purpose, we have built a simple dynamical systems model of this circuit and used that to drive the migration of an agent up a in silico sensory gradient. So with the full model, the agents indeed behave like real animals, and they were able to efficiently migrate up the sensory gradient, and in that process, alternating between the roaming and dwelling states. In comparison, we also adopted a model where the sensory input is only directed onto the circuit node that drives roaming. And the agents controlled by this reference model is less efficient at aligning itself with the gradient direction. So what gave rise to this difference? It turns out that the agents that are driven by the full model can quickly switch from roaming to dwelling when they start to deviate away from the gradient direction. Whereas for the reference model, there's a much longer delay for this state transition. And at the circuit level, we think this happens because with the full model, for the full model, the saddle node bifurcation occurs for higher input levels than for the reference model. And what this effectively means is that as the input starts to drop, when the agents start to deviate away from the gradient direction, the bifurcation happens earlier for the full model, and thereby opening up a larger region in the phase space, which allows the circuit to transition rapidly from the roaming-like state to the dwelling-like state. So altogether, we think we've identified a circuit where it's able to attain persistent activity state through mutual inhibition, and it promotes rapid state transition by directing top-down sensory drive onto both sides of the mutual inhibitory loop. And with that, I would like to thank my great team, and I will take your questions.
Questions? So I had one. Your CNN identified AIA as this neuron important for transitions, but is there any evidence that it might be a pattern of activity across multiple neurons that most effectively drives these?
So we indeed think that it's actually a multi-neuron activity pattern because taking AI away doesn't complete disrupt the ability of the network to predict transitions. So we think there's a combinatorial pattern, but experimentally, at this point, we only manipulate AIA by itself. Yes.
Okay.
So I didn't quite get how, so AIA is like giving like excitatory inputs to both like 5H like serotonin neuron and PDF, right? Like and like these like because of this mutual inhibition, like these serotonin and PDF neurons are like one high, one low state, right?
Mm-hmm.
I like
Yes.
do you have like any intuition how like both excitatory inputs, like shared excitatory inputs, can like flip back and forth between two states? Is there?
Yes, I went through that part very quickly, but the key is for the two outputs from AI to be asymmetric, so that the serotonin and PDF part of the circuit actually are sensitive to different range of AI activity. So actually, so we did parameter screen to see what's the set of parameters that make the full model perform the best. And it turns out that we need to have essentially the serotonin neurons become more sensitive to low range of AI activity, and the PDF neurons are more sensitive to the high range of AI activity or sensory input.
Thank you.
So, yeah.
Other questions? Okay, let's thank me again. And see you at the posters.